Hi everybody. The topic of my final project is personal genomics profiling. So what is personal genomics profiling? These days you can easily obtain your personal genomic profile using any of the commercially available services. There are also many research papers that are being published showing links between genes and diseases. So this project is an attempt to see if we can use the publicly available data to mine and get some insights into our health, our own personal health. So how did I go about it? The technology stack I used is what we have been using in this class. It's a Windows 10 machine which is running a VMware workstation. I used it to create a CentOS machine with Spark, Java, Maven, and Eclipse. The programming languages that I've used for this particular project is Java, Spark, and a D3 JavaScript library, which I have used to create HTML visualization files. So the data sets I have used is primarily from Genome's Sample Resource Project. This is a project which has personal genome data for thousands of participants. So I have taken one file from there. Basically, this file shows all the genes present on chromosome 14, along with whether they are present in any of the biosample IDs, as you can see here. The second file I have used is from the same source. This is kind of a complementing file to the first data set, wherein you can find the gender and ancestry details for the samples present in the first file. The third file I have used is from DigCNet. These they are they provide a data set which has the gene and disease association. They also have they have curated and all variant versions for of it. I have just used the all variant version in my case. So how did I implement it? So basically I created a Java application which on a high level does these things. It parses the file from IGSR to find the gene variants present in a single person. Then it cross-checks that those gene variants with disease association data from DigiNet, and then after filtering and creating the filtered data, we use these data in an HTML using the template approach. So what I have done in this case is basically I've created a template, a template-based HTML file, and which have tokens in there. And then when my program runs, I get the data and replace the tokens with the actual data I get from the Spark program and create a new .html file. So this file is a single file, and it will show you all the details regarding the um, visualization in a single file, so you don't have any other packages involved. How do you deploy and install it? This is basically a typical Maven project. So you use Maven to create a jar file, and you submit that jar file to the Spark engine using the Spark submit command. So see these are some of the example outputs that I generated from the file. So you can see I will show this again when I do a walkthrough of my code. But on a high level, the blue big circle is a person. These small gray pinpoints are the genes, and the red red circles are the diseases. And as you can see in this case, a single gene is kind of affecting and causing an influence on all these many diseases, and that's some of the interesting insights I got from doing this project. So I think this is a good point where we can step and walk through the code. So let's go right in. So we this is the Java project that I created, and we So this is the structure of the project. There is a source main Java class, and we have three Java classes that we created. The app.java is the main class here, and it also has the main defined in here. So we just create a simple Spark session. We are getting three parameters from the command line. The first one is the sample ID itself. So this is the biosample ID that we will use to filter out all the data. So the second line, other two parameters that we get are the input directory and the output directory. So we expect certain inputs in specific formats, and we expect all these inputs to be in our input directory. I will give a brief overview of all the input files that we have in this directory. And the output directory is where the program will run and generate out the .html file that we can play around with. So the second line is basically just loading the data set which has the gender and ancestry details. This line loads in the genome file, which this is a really big file. So I had initially wanted to do it for all 26 chromosomes, and just one chromosome's data is around 20 GB, and it did not have enough hard disk space in my machine to do this. The third file is a SNP disease mapping. So this is the file which gives you a gene and a disease mapping data set. So first you filter the info data to just the sample that you sent in the command line. Then you create a distinct. So one thing I found out was this data set had multiple associations, and there is a lot of use case from that too, because every association is a different research paper. But in our project, to limit the scope, I just did a distinct between a gene and a disease. And then you filter. So the genome data basically has the user ID as a column. So you filter the gene and the user that you're sending, and because these are the only two columns that you're interested in, and you filter also based on the data. So if it is 050, that means the gene or a gene variant doesn't exist in that sample, so that we can only get the genes that are active for that person. And then we join the filtered dataset that we got with the disease mapping dataset, so that and find every gene which has a disease mapping. And then we use the transform function to create a person SNP class. This The only reason I did it is because this way it is easier to send the file. When we create the JSON, the properties are set perfectly if you have a object to set your data into. And then after generating all this data, we send it to our report generator code. This basically takes, again, you get all your JSON and create a list of that, which you can then use to print into our HTML, and then we just get the individual ID, the sex, the population, the superpopulation name of the person, and the output directory where all this has to be printed to. And then, so if you look inside this code, you will see it's a simple report generator which creates a report from template. It reads a report.template file which is present in our input directory HTML template directory, and it just replaces all these tokens which are available in that template with the data that we just got out from our Spark file, and then just writes it into a file and closes it. There's nothing. Another thing of interest to see would be the dependencies. So if you look at the dependencies that we had to set for this one, it was the three Spark libraries: Core, SQL, and the Hive. Let's see a running sample of how this project works. So let's just build our project first. The other hand, while it's building, let me show you the input directory that we have. So So this is our input directory. The first one is the all variant is the file that we have from the DigiNet, which shows you the associations between diseases, and so the SNP ID head SNP ID, that is the gene ID, and this is the disease ID. But I have used the disease name, which is somewhere around here, as the mapping data that I was interested in. The other one that we have is a detail.tsp. This, as I explained, is just a simple, data set about what is the gender of the person, what is the population or code of that person. And the third one that we should be looking at is the raw files. So the reason I set it up like this was, initially I wanted to process, as I explained, I wanted to process all 26 chromosomes. So my idea was all the chromosomal files will exist in this raw files and the program can just process them. But as you can see, just a single file is around 27 GB, and I did not have enough space on my database to do this. So if you look at this file, it is a very big file, and uh, maybe I should just show you the header. So if you see the header here, you can see the first couple of columns are so first column is the chromosome number, the position in that of the gene in the chromosome, and then the ID. And this is the ID that matches to the SNP ID. And from here, all the column headers are the different individuals. So this is the setup for the data that we are trying to process. And as you can see, just the header spans like multiple page sizes. And the other interesting file here is the HTML template. So if you look at that one, so what I have done here is basically this is a fully built HTML file with certain tokens for the data set as like at the rate. So my Java program, when it runs, it knows to look for these tokens and replace them with actual data. So we have tokens for name, gender, population, and the same thing exists for these functions. So what happens in this function is basically we already, because we are JSONifying the person.snp that we created here, and as you see, uh, so we already have these three properties in there: person ID, SNP ID, and disease. So in this case, what we're doing is making links based on that data. We are making person a node, SNP a node, and disease a node, and we are saying the source is person and the target is SNP, and then we are creating one more link, which is the source is SNP and the target is disease. And we are just going to be using SVG to create an HTML um, file using this data. So let's go back. So if we see, I think our project had completed building, and it generated a jar file at this location. So now we are going to run this in Spark. I believe I already have it in my somewhere. So this is my command line to run it. I pass the same jar that was generated here. This is my input directory, which has all the input files set up there. This is the sample ID I'm using. This is a person's randomized ID, and this is where I expect my output to be. So I'm just going to run this one. So it does take a little bit long because it's the 27 GB file, but believe me, it will be worth it. So we just generated this, and you will have in our output directory, we'll have a file that just got generated. So if we open this in Mozilla, I believe that's the ID that we used, HD3720. And here it is. This is a visualization that was created. As I mentioned, this blue circle in the middle is the actual stripe, is signifying the person, and these gray pinpoints are the genes, and these red um, circles are the diseases. So as you can see, so what we are trying to highlight is how a person has a gene, and then these are the diseases that are, according to current research, are associated with those genes. So as some of the interesting data is, as you can see, the same two genes are involved in the systemic arterial pressure and blood pressure finding. Another interesting gene is right here. You can see the single gene is impacting all these diseases. Um, so it's just some visual data that you can play around with, and hopefully makes you understand this data set much better than just seeing it in a Excel sheet or something. I hope you enjoyed this presentation. Thank you.